So this is going to be the first part of chapter 20. We're going to move into talking about biology. So you should be watching this on Wednesday and then watching the second half of chapter 20 on Friday. Okay? So we're going to start off talking about the nature of living things. So to start off, we're going to define what it means or what something requires to be defined as alive. So there are going to be 5 characteristics of life. They're here in black. Metabolic processes. These are chemical reactions, energy changes. Some examples are nutrition uptake, nutrient processing, waste elimination. So this is, your eating, your digestion, and then you're excreting the waste. Generative processes. This is growth and reproduction. Responsive processes, irritability, which is not being cranky, but it's your rapid response to stimuli, so touching a hot stove and very quickly removing your hand from the hot stove. Individual adaptation, so if you move to a colder climate, you eventually become adapted to the colder climate. It just takes time, so that's individual adaptation. Population adaptation is about a group. And because it's a group, the other word for it is called evolution. Control processes is the fourth, and these are the coordination and regulation of metabolic activity, so your body working together from top to bottom. And then structural similarities on a cellular level. So here are 5 images if you are a visual learner of metabolic processes, generative processes, responsive processes, control, and structural organization. So you see in the upper right-hand corner, meiosis, mitosis. In the left-hand corner, you see adaptation with bunnies, population, evolution. The guy's fixing to pop his hand with a hammer. Control processes, coordination, so being able to see a mouse and go down and grab it. Then you have on B, it's called regulation. So whenever you produce like a product in a biochemical pathway, it will go back and interfere with the first reaction and basically makes it stop. And so most of your biochemical processes are very similar to this A, B, C, D product regulation. Structural organization, you have both on a large level, like your entire body system, circulatory, respiratory, but then you also have an organization on a cellular level. So early cell theory is what we're going to start with. Anton von Leeuwenhoek in 1632 to 1723 was his lifespan. He was the first to see what we know as a true individual cell. He called them animalcules because he didn't really know what they were, like small animals. Robert Hooke in 1635 to 1703 is his lifespan, studied thin slices of cork. And so you can see his image right here on the bottom right-hand side. It was a piece of a cork tree, a branch. Excuse me. You drink water. So the same thing the cork is made out of. And he termed those individual little black structures there that look like bricks, he called them cells. So he was the first one to really coin the term cell. Later on, there were 2 types of protoplasm later identified. The cell nucleus, which is a central body within the fluid, and the cytoplasm, which is the fluid portion of the protoplasm. So we have the cell nucleus, which is on the inner side, and then everything between the nucleus and the outer membrane of the cell is going to be cytoplasm. There are 2 major cell types. The first is prokaryotic. These are going to be cells that have no true nucleus. Bacteria and archaea are the 2 groups that are defined as prokaryotics. So pro will always mean no true nucleus. Eukaryotic cells have a true nucleus and other membrane-bound organelles. And so the examples there are algae, protozoa, fungi, plants, and it should be plants, animals. And so you see here on the on the left-hand side, you have a prokaryotic cell, which is a type of bacteria. And then on B and C, you have examples of plants and animal cells. Now, all cells, no matter what, have 5 common traits. So it's not the nucleus, because we said that some of them will have it and some of them will not. But the first common trait is that they're all contained within a cell membrane. All the material, all the matter is contained with a cell membrane. They all have some sort of cytoplasm, the fluid in which the cell contains inside the membrane. They have genetic material, which is necessary for reproduction and passing on genes, whether it's mitosis, meiosis, or asexual reproduction. They're going to have energy transfer molecules and then enzymes and coenzymes, which are important for biochemical processes. All right, so cell membrane. Cell membranes are thin sheets shaped into different structures and forms related to the function of the membrane. So they're going to be a little bit unique. So they're mainly made up of phospholipids and proteins. Phospholipids are polar molecules, so they like water, that form what's called a double layered sheet or a phospholipid bilayer, and with water on one side and water on the other. So if you look at this image on the bottom right hand side, there's the little red portions. Well, those are the parts that like water. And so the little red balls are going to point outside the cell, which is extracellular, and inside the cell, which is intracellular. The little yellow tails that are pointing towards each other like jellyfish, well, those don't like water. So they're going to point towards each other. That way they form this area where water does not like to stay or be. So it's kind of like the same thing as why water and oil don't mix. They're not miscible, they don't mix. Well, the little yellow tails don't mix with water either. And that's important because if the yellow portion liked to mix with water, well, there would be no reason for these little molecules to stack up the way they do. So if we looked at like a cartoon schematic, the bottom left-hand image shows us the lipid bilayer. So it's called lipids because these are little fat molecules. And they call it a bilayer because there's two layers. There's a top layer and the bottom layer. And you can see that it goes all along the cell. And within the cell, there are these proteins that are inserted inside different types. Okay. So we're going to talk about different types of motion or movement across the membrane. And we're going to start with diffusion. So diffusion is going to be the net overall movement of a molecule from a place of relatively high concentration to a place of low concentration. So it's like a ball rolling down a hill. That's a favorable process. It doesn't take energy to make that happen. Diffusion in this case is also called passive, which means it doesn't require energy. And our currency in the cell, the money of the cell, is called ATP. So when something is passive, it means it doesn't need ATP to happen. So it's not like if you think about going across a toll booth. When you go across a toll booth, you have to pay money, and that's the money to get across. The rate of diffusion is related to kinetic energy, the size, and the concentration. So how fast something moves across the membrane, it depends on how much energy it has, how big or small, and the concentration difference. The difference in the concentration across the membrane is called the concentration or diffusion gradient. And so you can see here in the bottom right-hand image, the blue dots. There's a lot outside the cell and only a few on the inside of the cell. So they're going to want to move across the membrane going down towards the slide so that there's an even number of blue dots on one side as there is on the other. Okay, so that's diffusion. Dynamic equilibrium is when molecules are being equally distributed and movement is equal in all directions, meaning if two molecules move across, then two are going to go back. Okay, so it's kind of like trading partners. You don't just, if you were thinking about like trading team members or trading, trying to give it a good example. Well, say that you're playing a card game and every person has to have 7 cards at each time and someone steals one of your cards. Well, you have to give them one of your cards so that everybody has 7. And that's dynamic equilibrium. If you take 3 of their cards, then you have to give them 3 of your cards. That way they have 7 and you have 7. And so we call it no net movement because everyone's maintaining 7 cards at all times, and therefore there's no concentration gradient because everybody has 7. And that's what dynamic equilibrium means across a membrane. It means if one molecule moves outside the cell, well then another molecule has to move inside to maintain that equilibrium number-wise. Now osmosis, on the other hand, is net movement. So it's the same thing we just talked about, diffusion, but it's specifically of water molecules. So we're not talking about trading cards like actual objects or molecules, but we're talking about water moving. So water is going to move through a selectively permeable membrane separating two solutions. So that is the phospholipid bilayer. Okay, it's selectively permeable. Some things can pass through and some things can't. So, but in this case, only water is moving. So passive movement is going to be from an area of higher concentration to an area of lower concentration. So water is going to move to essentially dilute the solutions and make them equal. So when something is selectively permeable, only certain molecules can pass through. They're going to depend on solubility, size, and ionic charge. So the amount of water in a cell is critical. If you have too much or not enough water, different things can happen. So too much water can dilute the cell contents and interfere with chemical reactions. Too little water can cause a buildup of waste products. So when we have what's called an isotonic solution, this is where we're going to have water and dissolved mineral concentrations inside and outside the cell that are equal. Okay, so iso usually means same. Okay, same. Tonic, in this case, we're talking about concentration. When we have a hypertonic solution, we don't have enough water. When we have a hypotonic solution, we have too much water. Okay, so hyper usually means more. So in this case, we have more solutes. Hypo usually means less. We have less solutes. But the relationship between the solutes and the water are inversely related. If I have too much solute, I don't have enough water. If I have not enough solutes, I have too much water. Okay, so what's going to happen when we have a selectively permeable membrane, and we're talking about osmosis, if we were talking about diffusion, specifically these yellow hexagons, which are sugar, would be moving. But in this case, the sugar molecules can't fit through that gap. Only the water can. So in this case, we have 4 sugar molecules on the left and 1 on the right. And what's going to happen is we have a 60%, 40% water-sugar mixture on the left and a 90-10% water-sugar mixture on the right. So in order to balance that out, the water is going to move to the left to increase the amount of water on the left and decrease that relationship between the water and the sugar. Okay, so that's what osmosis is. Diffusion would be the sugar moving. Osmosis is the water moving. So cells are going to be affected by the amount of material and water that they have. So when we have an isotonic solution, you'll see on the upper image, these are red blood cells. On the bottom image, these are plant cells. So red blood cells are normally like flattened discs that kind of have like a point pushed in the center, like little frisbees or donuts. And this is what they would look like in an isotonic solution. Plant cells would be square, just like you see here. Now, when we have a hypotonic solution, if we look back to what we said before, in a hypotonic, we have too much water. So when we have too much water, the cells are going to swell and they're going to rupture. And so you'll see that the red blood cell here is bursting and the plant cell is very, very swollen. When we have hypertonic, so let's go back, hyper, I have not enough water. So in a hypertonic solution, the cells are going to shrivel up, and you can see here that the cytoplasm in the plant cell shrinks away from the cell wall. So even though it's a square shape, the cytoplasm on the inside shrivels up just like the red blood cell does. All right, facilitated diffusion. So now we're going to talk about a diffusion process. So it's still passive, but it's going to be where molecules move across a membrane according to the concentration gradient, so still abiding by diffusion, but they're going to combine with a specific carrier protein. So this is like the groom carrying the bride across the doorway for the first time. The groom picks up the bride and carries her through the door, and that's what specific carrier proteins do. They pick up the molecule from one side of the membrane and they carry it across the doorway or the membrane to the other side. So this allows molecules to travel from an area of high concentration to low concentration that wouldn't go through diffusion by themselves, meaning like this sugar molecule, it's too big to cross the membrane because it can't fit through the hole. However, through facilitated transportation, it could if it had a protein to carry it across. So in this case, passive transport, only energy is required, no chemical bond energy, no ATP. And so if we look down here on the right, the orange triangles, they are able to move across this purple, or the membrane through this purple channel, and that is called a membrane protein. So their only requirement here is no ATP, but there must be a carrier protein to do the job. So it can't just be any old protein, it has to be a specific kind. So let's go back to looking at this image. If you look back on this first one, you see where it's got on the left-hand side, the first two purple proteins, they are called membrane proteins, and these are ones that will help to carry certain molecules across in facilitated diffusion. Okay, so it's like facilitated living. There are people there to help you live, and these are things that are there to help move things across. Now, active transport is kind of the last kind we're going to talk about that are in this diagram on the bottom right. So this is where molecules move from an area of low concentration to high. So this is the opposite of everything else we've talked about. This is moving the ball up the hill. To do this, cells must expend energy, and that energy is called ATP, adenosine triphosphate. It breaks down into what's called ADP plus phosphorus plus energy in red. It's a very specific process. Only certain molecules or ions can even undergo this, and there still must be a specific transport carrier protein to do it. So it can't just be anything. It has to be a specific protein, a specific molecule, but ATP is required. So looking at the bottom image now, the green blocks, you see that there's only 2 green molecules on the outside of the cell, and there's several, probably 12 on the bottom right, so inside the cell. Normally in diffusion, the molecules would be moving up. The green blocks on the bottom would be moving up to the blue side of the cell, which is outside. But because of ATP, we're actually able to basically transport these things against the gradient, which means moving them inside the cell. Now, phagocytosis is a process in which a cell wraps a membrane around a particle, could be food, bacteria, virus, or foreign molecule, and engulfs it. So it's eating. That's what they call it. It's kind of like, for better or less words, cell eating. So you can see here we have a cell on the left, and it's opening up and wrapping around these microbes, eating it. It becomes ingested, it goes through a process, and then there's exocytosis, which is like the equivalent of pooping for a cell. And so then on the right image you see also it's kind of taking these little red pill-like things and eating them. Okay, so that's phagocytosis. All right, so we're going to pick up with the rest of this lecture in the next part of the video, which you'll watch on Friday.